Hello everyone! So this is going to be the video that goes through the steps of respiration that you need to know for this class. It'll also go through the diagramming part of the poster we do. So just keep in mind this video is an oversimplification of the reactions that occur. We are focusing on major inputs and outputs of each part of respiration. So if you look at other resources online or other videos even, it may include stuff we did not talk about. This is the fundamentals you will need to know and be able to demonstrate on your FRQ and test. So just keep that in mind as we're going through this, and if you use a different video to help you study. Okay, so here is our mitochondria. So we know the mitochondria is the powerhouse of the cell, basically meaning it's able to perform cellular respiration. Inside our mitochondria, so inside both of the outer membranes, we have the intermembrane space, and then on the other side of this very inner membrane, we have an area referred to as the matrix. So we'll be referring to both those areas as we go through the reactions that occur along the way. Another thing I want to point out just really quickly is the summary equation for respiration, so that way we can look at it as we go through. So we'll see our reactants in the summary equation are C6H12O6, glucose, plus oxygen yields carbon dioxide, water, oop, that's a 2, okay, sorry about that, water, and ATP energy. And ATP energy is going to be our ultimate goal of this whole series of reactions. We are doing this entire process to make ATP. All right, so there's our summary. There's our parts of the mitochondria. So now we're actually going to get started with the reactions. And we'll see the initial reaction of respiration actually doesn't occur in the mitochondria at all. It's going to be called glycolysis, and it actually occurs in the cytoplasm. So glycolysis is our first step where we are able to take glucose in its very first form, and we are going to start breaking it down. So please remember that glucose is a 6-carbon molecule, and the way we're able to uptake energy from glucose is from the bonds between the carbons, and also it has high energy electrons within its structures, also the bonds, right? So we have glucose, which is our energy molecule we're going to start with. And we'll see glucose, which is a 6-carbon molecule, is going to be broken down by several different enzymes that you do not need to know into 2 pyruvate molecules. Now, pyruvate is going to be a 3-carbon molecule. So we see ultimately what happens here is that we took a 6-carbon molecule and we basically broke it up into 2 3-carbon molecules. And through breaking that up, we're able to generate some ATP, not a lot, but some. And we're able to fill up one of our electron carriers. So we'll see NAD+ will come by, and it's able to pick up 2 electrons and a proton from that glucose breaking down. So ultimately, glucose is being broken down into 2 smaller molecules that have 3 carbons each. That process is generating some ATP and a little bit of NADH. This NADH, we will see, comes into play later at our electron transport chain, so I'm just going to show that it's going into this matrix space. Okay, so now I'm just going to continue. So that's glycolysis, that's our first step. We'll see after our pyruvate is made, it is going to enter the mitochondria and actually go to this matrix space. And within this step, we'll see 2-pyruvate, again, 3-carbon molecule, will get broken down slightly again into a molecule called acetyl-CoA. And acetyl-CoA, we will see, actually only contains 2 carbons within it. So what's happening in this step is one of those carbons on each of those pyruvates are being removed. And we'll see the carbon that's being removed is going to be in the molecule CO2. So it's released as CO2 in this step. So we're going from 3 carbons to 2 carbons, and that carbon that's being removed in that process is CO2. And it'll diffuse out. Okay, and in this process, again, every time we're breaking it up, we're getting some kind of energy. We are making more NADH in this step, and it, just like last time, we'll go to this final step, we'll get to it. And an H plus is made every time this happens as well. And we'll see again, those H pluses will come into play in our final step. All right, so the next step is going to be the Krebs cycle, which also occurs in the matrix. So we'll see acetyl-CoA goes into the matrix where the Krebs cycle is going on. And basically what's happening in this step, it's the final step where our carbon-based molecules that originated from glucose are being used for energy. So we're breaking that final bond between our carbons and acetyl-CoA, and we see when we break that bond, we are able to make a little bit of ATP. We see NADH and FADH2 are able to come in and take their electrons and protons. And then we'll see what's left over after the energy is taken from our acetyl-CoA carbon molecule is carbon dioxide will be released. That'll be kind of the leftovers. All right, so the final step of the entire thing is going to be chemiosmosis, which contains the electron transport chain. So what we've been doing all along is we've been charging up our electron carriers as we break down glucose. So now these electron carriers are going to come into where the electron transport chain starts. They're going to drop off their electrons, and they're also going to drop off their protons that they've picked up along the way as well. And we'll see the whole purpose of these electron carriers are so our electron transport chain is able to have energy as electrons get passed through this network of proteins. They're going to have energy in order to do active transport. So we see these H pluses are able to be transported actively across this membrane. Once the electrons go all the way through the electron transport chain, they become released. So this is where we need oxygen. Oxygen's role is to come in, pick up these electrons, also pick up some protons, and form water. So now we have our whole reaction at top pretty much completed. This last step is just to make the majority of our ATP energy. So these electron carriers, their job is for the active transport, powering those proton pumps to be able to move the H pluses. So what we'll see is now those H pluses are moved actively into this intermembrane space. They want to get out and space out. They don't all want to be concentrated in this area. The only way for them to get out of this area is by traveling through ATP synthase. And the force of them pushing through ATP synthase is going to give the enzyme energy to be able to link together that third phosphate onto ADP. And this step is going to make the most ATP out of the whole process of cellular respiration. So I'm going to draw a lot of ATP here so y'all can see that. You say ATP. It's kind of hard to see. And that's where most of the energy is actually coming from, generated from respiration. So this whole process, you can just see how glucose is being broken down, what it's turning into, what it's being used for, and then this final step, which is using all of that cumulative electron carriers that are being charged up to power ATP synthase, which is able to make a lot more ATP than any other step.